All right, so good morning, good afternoon, and good evening, everyone, and thank you so much for joining us today. The following presentation is brought to you by nonprofit organization iExplore. I am honored today to be presenting to you our seminar presenters today, Wendy, Andrea, and Dorothy, who will be giving us an in-depth seminar on inheritance and genetics. So, guys, you guys can, time is yours.
All right, hello everyone. Welcome back if you were here for last time. This seminar is of course gonna be taught by me, Andrea, and we have a guest speaker today, Dorothy. So without further ado, I guess we'll just start. Next slide, please. All right, so the contents we will be covering today is the concepts of genetics and inheritance. We'll have a little review on alleles, and we will go into a bit more complex alleles. We'll of course do our Punnett Square review again. And then we're gonna talk about blood types and how you can understand the blood types and how genetics basically affects it. And it will transfer to Dorothy, who has a whole new slide prepared for us. All right, so we're here on our first section, the concept of genetics and inheritance. So, importance of genetics. So genetics is the science of heredity, which basically is just, heredity is just about us learning about how parents pass on genes and traits to the children. And why that's important is that it helps us understand how or why we have certain physical traits like phenotypes. And it also helps us understand how diseases are passed on, which really helps preventing a lot of diseases. And the more you understand diseases, the easier it is to cure it. So next slide, please. So the field of genetics. The study of genetics revolves around understanding how traits are passed down from parents to children. Like I said before, this is basically the study of genetics is just heredity. So jobs that include genetics and what you will use genetics in are biochemistry, medical science, biochemical engineering, and forensic science. So we will go into Mendel's principle of independent assortment first. So each pairs of alleles segregates independently during gamete formation. This means that each of the alleles for each specific trait are not associated with each other. So what alleles means, hopefully you guys remember from last time, alleles are just different versions of genes. So basically, each trait have two alleles determining the phenotype, and each trait's allele is not associated with each other. So here we have an example. So the two alleles determining for your hair color and the two alleles determining your eye color will not affect each other. So here we have the first question. This is really all of these questions right now will be Punnett Square related, and so if you guys can just read this and then solve it using a Punnett Square. If you don't understand, that'll be okay because we will go over basic Punnett Square again later on. So remember that Mendel's peas have purple flowers as dominant and white flowers as recessive. If Mendel breeds a homozygous dominant pea with a homozygous recessive pea, what are the chances of the F1 generation of having white flowers? So I'm gonna start the pose, and it's okay if you don't understand because you guys will understand later on. So I'll give you guys a couple more minutes. All right, so I'm gonna end the polling right here since half of the people already did it. So most people picked 25%, which is actually false. It's 0%. So remember, dominant alleles, when paired up with the recessive allele, will always cover it up. And the only phenotype that's gonna show up is the dominant trait. So if you breed a homozygous dominant P, which means both of the alleles for the P will have purple flowers as their trait, and a homozygous recessive P means both alleles will be for white petals. If you use a Punnett square, you'll find out that all four of the offspring will have capital P, lowercase p. And since, as I said earlier, when a dominant allele pairs up with a recessive allele, the dominant one will always show. So that is the case for all four of them. So having a white flower in the F1 generation is impossible. And that's why it's 0%. So can we go to the next slide? All right, so Andrea will be teaching you guys about alleles.
Okay. Hold on. Wendy, how do I move the poll question to the side because it's kind of...
Oh, just exit it out.
Oh, okay. So let's talk about alleles. For the following slides, we will use the example of Mendel's experiment with pea flower color. And the alleles that will, the letters that will stand for these alleles are capital P for purple flower and lowercase p for white flower petals. So, homozygous and heterozygous alleles. For each characteristic, an organism inherits two alleles, one from each parent. They can inherit the same form from each parent or different forms from each parent. And the two different forms, and the two types of allele pairs will be homozygous, which are the combination of two identical alleles, or heterozygous, two different alleles. And then the next set of information about alleles will be phenotype and genotype. So the definition of phenotype, as said in the previous seminar, is physical characteristics, and genotype meaning genetic characteristics. So phenotype will be what shows up in the organism physically and what you can see with the naked eye. And genotype is the alleles, basically. The phenotype of the homozygous genotype, capital P, is purple flowers. So with this type of genotype, the flower will show up as purple. The phenotype of homozygous genotype, lowercase p, is white flowers, which means with this allele pair, the flowers will be white. And then the next set of information for alleles is dominant and recessive alleles. So a dominant allele always has a capital letter, and in this situation, it will be capital P. The trait that is expressed in the phenotype is the dominant. So if an allele pair has a dominant allele, the phenotype that shows up in the organism will be the dominant. The recessive allele is always lowercase, and in this case, it will be lowercase p, standing for white flowers. And this is the trait that is masked by the dominant allele. So in conclusion, an allele is one of two or more alternative forms of a gene. It will be uppercase P for purple flower color, lowercase p for white flower color in this situation. They are inherited in pairs that may be heterozygous or homozygous. And lowercase is recessive, and uppercase is a dominant allele.
Okay, so we have the second question. So hopefully that little section clears some things up. And so remember that Mendel's pea have purple flower as the dominant variation and white flowers as the recessive variation. If Mendel breeds a homozygous dominant pea with a homozygous recessive pea, what are the chances of the F2 generation of having white flowers? So what we need to understand here is that what's different from the last question is that this question we're no longer talking about the F1 generation. So the F2 generation are basically the children of the F1 generation. So what we're really looking for is what happens when you cross one of the peas from the F1 generation with another. So right now I will launch the poll and you guys just try your best. All right, I'm just gonna end the poll right here since most of you got the question right. It is 25% because remember from earlier, the F1 generation we mentioned got all of them have capital P, lowercase p, which means they have one allele representing the dominant purple flower and one allele representing the recessive white flowers. So if you put them into a Punnett square and you calculate it, one of the squares should be a homozygous dominant square with both capital P. Two of the squares should be heterozygous, which should have a capital P and a lowercase p. And lastly, the last square should be homozygous recessive, which having both of the lowercase p and be white flowers. So as I said earlier, dominant alleles cover up the recessive alleles when they're paired up together. So both of the heterozygous one will also be purple. So since 1 in 4 of them is white, it will be 25%. So hopefully that clears things up. So next slide, please. All right, so now we will move on to the more complicated alleles or the more complex alleles. So in additional parts to allele pairs, not all allele pairs follow the basic rules mentioned in the previous section. They're not all just dominant recessive, there's codominance and incomplete dominance. And we will learn about additional components with the blood types as examples. So codominance, some alleles are always expressed even in combination with other alleles. Heterozygous display phenotypes, both of homozygous phenotypes and codominance. So what this means is that previously mentioned heterozygous traits phenotypes will always show as the dominant trait because the dominant one covers up the recessive one. Some traits will always show. This is what we call codominance. So if we take the flowers as example, let's say white instead of recessive is codominant with purple. When we have a capital P and a lowercase p together, instead of being purple, the flower will be both purple and white. Next slide. So let's have another example, human blood group alleles. Allele A and B are codominant, like the blood types A and B. So once in a Punnett square, we got both A and B in one square. They're paired up together. Instead of having either blood type A or blood type B, we'll have blood type AB. And this is what happens when the individual had the genotype of AB. All right, so we have another question. Assuming the black fur and the white fur on a type of mouse are co-dominant. So this time we will be utilizing the information we just learned in the last two slides to answer this question. So assuming black and white fur on the type of mouse are co-dominant, the father is a homozygous dominant with black fur, and the mother is homozygous dominant with white fur. So what are the percentage of their children with the phenotype of both black and white fur? So I'll go ahead and launch the post again. Okay, so since some of you already voted, I'm just gonna end polling right here. So unlike most of you who said 50%, that is not correct. The correct answer is actually 100%, because remember, both of the traits are they're co-dominant, which means if you put them into a Punnett square together, they will both show. So when they will both show, the children will have black and white fur. So for the children to have black and white fur, the genotype must be like have an allele for black fur and an allele for white fur. And since both of the parents are homozygous, that means all of the children will have that following genotype.
So next we will review the basic Punnett square. So this is the chance for those of you that don't know what a Punnett square to learn more about it. So what is a Punnett square? It is a square diagram that is used to predict the genotypes of a particular cross or a breeding experiment. So basically, the parental alleles are placed at the top and the side. So just like this. And then after you place the parental alleles on the top and the side, you will take one allele from the mother and then drag it down into the correlating box and do the same with the other parent. And then just like this. So after you finish doing the same for the rest of the 4 boxes, you will predict that 1/4 of the offspring are homozygous dominant, which will give a purple phenotype, meaning the flower will be purple. And then 1/2 or 2/4 of the offspring will be heterozygous dominant with a purple phenotype, meaning the dominant has masked the recessive allele, and the flowers will be purple. And 1/4 of the offspring are homozygous recessive, meaning that there are no dominant alleles present in the allele pair. And this will result in white flowers for the offspring. So overall, a Punnett square allows you to calculate the percentage of offspring that will inherit certain genotypes and phenotypes. So this can help you predict how many of the offspring will look a certain way or have a certain trait.
Okay, so we have another question here. Assuming that the fluffy fur is dominant and the silky fur is recessive in wolves, if the mother is homozygous dominant and the father is heterozygous, what are the percentage of the children having silky fur? So this is just like every other Punnett Square question. I will, of course, launch the polls for you guys to. So I'm just gonna answer the question while the poll is still in progress, since not everyone have voted. So let me explain. If the mother is homozygous dominant, assuming that I'm assuming that all of you are doing Punnett squares right now. So assuming that the mother is homozygous dominant, we will use the letter P again for this example. We will have both capital P for the pairs of the mother because dominant alleles are represented with capital letters, and homozygous means both of the alleles are the same type. So, and to the side, the father is heterozygous. So what heterozygous means that both alleles are different. So since in the last paragraph we read that there's only two types of variations, that means the father will have to have a capital P and a lowercase p. So now that we have both of the parental pairs of alleles, we can drag them down. So if you drag everything down, the first box should have two capital P from the mother and the father. The top right box should have a capital P, lowercase p, and that would be the same for the rest of the boxes. So this is because the mother is homozygous dominant, and because dominant alleles will cover up recessive alleles when they're paired up together, and there's a dominant allele in every single box, there's 0% of chances for the children to have silky fur. And that's it for the question. Let's move on. Okay, so finally we move on to blood types and how genetics affect blood types. Let's learn like the basic things about blood. So a person's blood type may be type O, type A, type B, or type AB. Matching the right blood type is critical for blood or organ transfusion. That is because if the blood does not match, it could possibly cause the blood cells to clump together, and this could possibly kill the recipient. So, unlike everything else we learned so far, blood types actually have 3 different alleles. So, if you guys probably can't see, but that is an I with a little a on top, an I with a little b on top, and a lowercase I. So, as I mentioned last seminar, that these the letters could be whatever you want them to be for if you're calculating a Punnett square, but that is different if we're calculating blood types. So the capital I with the little a is type A blood, capital I with little b type B blood, and lowercase I is type O. These are this way because type A and type B are co-dominant, while the type O is recessive. So can we move on? All right. So yeah, as I said earlier, type A, type B are co-dominant, and type O is recessive. So blood types can also be determined by a Punnett square, just like every other trait. Okay, so here we have a little diagram for you guys. So both type A and type B blood are co-dominant, so they will both be expressed in the phenotype. So the reason that blood will get clumped together if your blood types is not right is because of the antibodies, which you will learn later on in Dorothy's slides. All right, so in order to get type A blood, since it's dominant, any expressed genotype, you could have homozygous dominant, which both are capital I with little a, or a capital I with little a and I, and B is the same but with the B. And to get AB, you have to have capital I with A and capital I with B. And to get O, you will have two lowercase I. So the antibodies present in blood. So O will have antibody A and antibody B. That means it cannot receive blood from people with type A blood type B, and type AB. And type A have anti-B body, so it cannot receive from B or AB. B cannot receive blood from A or AB, and AB can receive blood from everything. So that is why AB is called the universal recipient, because it can receive every single kind of blood. And O is called the universal donor because as you can see here under the O line, everyone else can receive type O's blood. Move on. So I believe we have another question here. So this one is gonna be a lot harder than every other ones we encountered so far. So just try your best. Elisa is considering getting a blood transfusion from a donor with type B blood. Her mother is a heterozygous with type A blood, and her father is homozygous recessive. What is the percentage of chance of a negative reaction / blood clot? So there's a lot of information in these few sentences, and I will of course launch the poll like always. So just try your best, even if you don't know. Okay, so I'm gonna go ahead and do the Punnett square with you guys. So her mother is a heterozygous with type A blood. So what this tells us is that the mother's genotype, which we will put on top, will be capital I and an A with a lowercase I. So, and her father is homozygous recessive. That tells us that he has both little I for both of his alleles. So if we drag it down, we should have two boxes with a capital I with an A and a little I, and two boxes with two little i's. So since she is getting a blood transfusion from a donor with type B blood, she cannot receive blood from this donor at all, because what we're calculating here is basically we're trying to find out what blood type Elisa is, so if she can get the blood type, the donor's blood. So it ends up she can't get the donor's blood type at all, because no matter what her blood type is, whether it's O or type A, she cannot receive the blood. If that makes sense. All right, so next slide. So right now we have a guest speaker, Dorothy. So she will be walking us through the bloods and just all of the information about blood. So we will transfer from this slide to her slide. So please hang out a little
I'll
bit.
stop sharing my screen now.
Yeah, so Dorothy, if you can share your screen. Since while she's getting her share screen ready, do anyone have any question at all? Like we can answer questions in this time. So do no one have any questions at all, or does everyone understand everything? We can still go back and explain some things if you don't understand.
Oh, actually, we got a question right here.
Oh, okay.
Okay, so someone asked, what is gamete and what is F1 generation?
Okay, so so the original parental generation that you start with is called the P generation. So once you crossbreed the P generation, which you get to the next generation, they are named F1 generation, and F2 generation, and F3 generation, and so on and so forth. So this is they're called like that because Mendel's purebred peas, okay, so Dorothy got her slides ready. I'll answer that later.
Okay, so hello everyone. I am Dorothy Huang. I come from Tianjin, China, and this September I'm going to fourth grade. And now we're going to talk about blood. See in this picture, there is a blood vessel and lots of blood cells firing through it under a microscope. Today we're going to talk about 4 parts of blood. Firstly, what is blood? Second, the components of blood. Thirdly, the blood groups. And last, blood vessels and circulation. And then we'll have a little review about what we've talked about. Firstly, what is blood? Blood is a part of the immune system. It is part of the circulatory system. And it's part of the circulatory system. How? Well, firstly, it's part of the circulatory system because, well, it circulates blood.
Okay, everyone, please don't draw on the screen right now. That is really distracting to our presenter and everyone in the call right now. So just please refrain yourself.
And it is part of the immune system because there are 2 types of cells in it, which have very big use in the immune system. And then it's part of the excretory system because it carries out respiration. Respiration is not breathing. Respiration is getting oxygen or nutrients and then excreting the waste materials. And the blood cells and the blood play a larger part in this process. Now we are going to talk about the components of blood. Blood is composed of 4 things: plasma, red blood cells, white blood cells, and platelets. Firstly, let's talk about plasma. Plasma contains hormones, antibodies, enzymes, glucose, fat particles, and salts. Hormones are like nerve signals. Hormones are like nerve signals that go through the blood. But they are way much slower than actual nerve signals. But they're also very important. Once they've said, they can function anywhere. And then there are antibodies. Antibodies just to the antigens that they are dedicated to. They can choose to smash the pathogens into smithereens, and this is an important immune function. Plasma also contains enzymes which help with digesting and other things like breaking things up. They also contain glucose, which is a type of sugar. And then there are fat particles, which are just breaking up fats, just like glucose, which is breaking up sugar. And sugar, fat, we've got 2 nutrients. And now we need another, salts. Plasma contains a lot of nutrients. And maybe that's why they make up more than half, or 54% of your blood. Now let's talk about platelets, also called thrombocytes. First, before their activation, platelets look like wrinkled up pieces of paper. But if a blood vessel wall is damaged, then it becomes activated and becomes all thorny. Then it goes to the part of the injury, and its thorns spit out a web, just like the spiders. But their web is not called a web, they're called a fibrin, which makes a blood clot, and no more blood can go out. Then it acts like a makeshift blood vessel wall. Now, we're going to talk about another type of cell, white blood cells. White blood cells are part of the immune system. They consume germs. How? Well, they cover the germs. They surround the germs. And then they turn the germs into pus. It's like they're great diners instead of police. They're like cannibals who kill and eat the criminals. But also, they do a good thing. They defend the body against infection. For if there are less germs, they will have less infection. And then there's a third type of blood cell called red blood cells. Red blood cells contain hemoglobin. Hemoglobin is a chemical which makes the red blood cells stick to oxygen. Why? Well, because they need to carry oxygen. Their concave surface makes them best for carrying oxygen. Also, if their oxygen is given to the body, then they will carry carbon dioxide. And if they're so important, why does the body make millions of them die each day? So they can make millions more? Don't know why, but millions of red blood cells are made each day, and millions of red blood cells are dead each day. Now, let's talk about blood groups. There are 4 main blood groups. There are A, B, and O. Blood groups are distinguished for transferring blood or blood transfusion. If there are no blood groups and everyone has the same blood, then there's no need to talk about blood groups because everyone has the same blood group. And if the blood groups are intertransferable, no one will discover their blood groups. So this is all used for blood transfusions. There are 2 types of antigens, A antigens and B antigens. A antigens are like very sticky things. And there are antibodies. Antibodies are also very sticky things. Antigens seem to call out to the antibodies. Just stick on me. And the antibodies stick on the antigens of their own type. See, the A antigen on this graph is round shape. So the A antibody must be round shape. The B antigen is arrow shaped. So the B antibody must be Y shaped. These are the 4 main blood groups. So you say, why cannot they transfer to each other? Well, it's because each type of blood contains antibodies and antigens. Type A blood contains anti-B antigens, which will stick onto the B antigens. And type B blood contains anti-A antibodies, which will stick on the A antigens. And this will cause all the blood cells to clump together, which ultimately results in death. And then there is also the rhesus factor. It is another factor of transferring blood. You see these plus marks and minus marks on the blood cells. These are actually determined by the rhesus factor. People with the rhesus factor cannot give blood to people without the rhesus factor. Most people, or 95% of the population, have the rhesus D factor, but the other 5% of people do not have the rhesus D factor. Maybe they lost it in some time of evolution. Now we're going to talk about blood vessels. There are 3 types of blood vessels: arteries, veins, and capillaries. Arteries carry oxygen-rich blood, which means that the red blood cells have a lot of oxygen in them. And veins carry oxygen-poor blood, which contain red blood cells without a lot of oxygen. Or you might say carbon dioxide-rich blood, because that is the waste product of the cells that they're going to take away. And then there are the in-betweenies between arteries and veins called the capillaries. These are where the oxygen is drawn out of the blood and the waste material is put in the blood. The blood vessels are like this. Big arteries branch into medium-sized arteries, branch into smaller arteries, branch into arteries, which eventually turn into capillaries. Then they come together to form veins, and bigger veins, and then bigger veins, and go back to the heart. Now we're going to talk about blood circulation. You see in this diagram, most of the blood goes to an organ in arteries, and then the oxygen and nutrients are drawn out in the capillaries, and then there are the veins which flow back to the heart. But the lungs are different. The veins go to them, and in the capillaries, the oxygen is drawn to them. and then the arteries come back from them. Why? Well, because the lungs itself are not muscles. They are simply air sacs which draw oxygen from the air. So the body's blood goes out in arteries, goes to the organs, turns into veins, comes back to the heart, and then goes to the lungs, becomes the arteries, and gains oxygen. Then it turns to the arteries again and go to the organs again and again and again and again. This is blood circulation. Now let's talk about our heartbeat. Firstly, the aorta, this biggest artery, brings the blood to the left atrium. And the vena cava, the biggest vein here, brings the blood to the right atrium. And then those atria contract and bring the blood into the ventricles. In the left ventricle, there's the oxygen-rich blood, and the left ventricle contracts into the aorta again, which goes out to the body. But in the right ventricle, there's the oxygen-poor blood, which goes to the pulmonary veins, which goes to the lung, which goes back to the heart, into the left atrium, which contracts into the left ventricle, and then goes into the aorta, and then goes to the body. This is complex, but it is blood circulation. Now let's talk about blood diseases. There are three main types of blood diseases: leukemia, anemia, or blood clots related disease. Firstly, leukemia. Leukemia is just like blood cancer. It is caused of abnormal bone marrow. Why? Well, because bone marrow makes all types of blood cells, and white blood cells get strange and turn into cancer cells. These just gobble up healthy cells instead of the pathogens that they're supposed to gobble up. And they also gobble up the healthy red blood cells. So, if the bone marrow gets strange, the white blood cells will get strange. And if the white blood cells get strange, the blood gets strange. And then there is anemia. Anemia is caused by abnormal red blood cells instead of abnormal white blood cells, or actually abnormal bone marrow. Anemia is caused by the red blood cells having a bit of genetical error, which then makes them lose hemoglobin or make them change shape into abnormal hemoglobin. This means they can carry less oxygen, and the body gets less oxygen. So the heart must beat faster to get all the oxygen to the body, and this causes more diseases. It also makes the people short of breath because they need to rest so fast. And then there are blood clot related diseases. There are firstly, I'm going to talk about excessive blood clots. They are normally caused by injuries to prevent bleeding, but turn out to be excessive. How? Well, the blood clots too much. and then it restricts the blood flow, causes pain and swelling. And that's bad, for if your blood cannot circulate anymore, you die. There are also not enough blood clots and zero blood clots. For not enough blood clots, it's because platelets have gone strange. And this way, the platelets just become less and less and less. And then there's not enough platelets to clot the blood. So only a small bruise may cause the body to bruise. And then the person will be very vulnerable to any cuts, but not as vulnerable as another disease called hemophilia. In hemophilia, hemophilia is passed down through the genes. If the genes turn out to be strange, the person gets hemophilia, a condition in which the person cannot clot their own blood. Then, if the person gets cut, it's very dangerous. Now, let's have a little review. Firstly, why is blood red? Although we didn't talk about this, I guess you could guess it out. Well, because blood is red because there are so many red blood cells in it to make it red. Second, what is blood made of? Easy question. We've all talked about it before. It's made up of plasma, white blood cells, red blood cells, and platelets. Next, where are blood cells made? As we talked about it when we talked about leukemia, blood cells are actually made in the bone marrow, not in anywhere else you might think, not in the blood, obviously. Next, why can a person with type A blood transfer his or her blood to a person with type B blood? It's simple. The antibodies in the type A blood stick on the B antigens of the type B blood, and the anti-A antibodies from the type B blood stick on the A antigens from the type A blood. So all the blood cells clump together, and it causes, well, death. Next, are white blood cells really white? Well, come to think of it, actually no. Red blood cells are only red because they contain hemoglobin, but white do not contain any hemoglobin. They do not contain hemoglobin, so they're actually see-through instead of white. They are only colored under a microscope to make you see their color, or actually to make you see them. Next, what is the difference between arteries and veins? Simple. Although there are lots of answers, arteries carry oxygen-rich blood, and veins carry oxygen-poor blood. Also, another thing is that veins contain valves to stop the blood from flowing in another direction, while arteries do not. Next, how do white blood cells kill bacteria? As shown in this graph, the white blood cell, or the macrophage, sees the viruses or the pathogens, and then it covers the viruses and it consumes them. Next, how does a scab form? As you might guess, a scab is just a very huge blood clot. It forms when the body is injured and the platelets come out and clot it. Then it slowly hardens into a protective scab while the other parts of the body heals. Last question, how do people get hemophilia? Well, it is very unfair to them because if their family history has hemophilia in them, he or she gets it. It's not because of smoking or anything. It's because of their genes. Any questions?
So that's all for today, and we hope to see you again in our future seminars. So bye, everyone.